Hello, my name is John Feller. I'm a 17-year-old junior at Canterbury School in Fort Myers, Florida. I work hard, study harder, and live my life in the face of Crohn's disease.
Last year, I did an experiment to try to determine a correlation between bacterial imbalances and inflammatory bowel disease, or IBD. This experiment was inspired by my previous experiment's ambiguity. For more information on this previous experiment, please see the section labeled 2012 to 2013 on my website. Since my data collection in this previous experiment could have been considered somewhat vague, I wanted to provide some more concrete evidence to support my first conclusions, as well as begin a second avenue of approach to the cause of digestive distress. These experiments have additionally persuaded me to hypothesize that these additions to our food are indeed a cause of digestive distress. Please allow me to explain. E. coli is known to be an important regulatory bacterium, meaning its presence determines the size and composition of other bacterial colonies that can be helpful in some quantities but harmful in others. Sampling has shown that E. coli populations in patients with IBD are typically off balance, and several artificial ingredients are known to cause flare-ups of symptoms in these same patients. In this experiment, I tried to determine if these foods cause symptoms by misbalancing bacterial populations and thus spurring an immune response in IBD patients. I thus asked the question, what effects, if any, do artificial additives have on the growth and development of E. coli? However, I did not stop here. Doctors sometimes recommend probiotics as a short-term alternative to counter IBD symptoms. Probiotics supposedly stabilize bacterial populations in the gut. Perhaps probiotics are a valid counter because they prevent or suppress imbalances in bacterial populations caused by food intake, as prior discussed. After a fair amount of research, I was able to hypothesize that, if applied to E. coli, additives would act to irritate said E. coli and thus limit and reduce colony size, while probiotics would counteract these effects. I then designed a valid experiment to validate or disprove this hypothesis. The independent variable would be the type and number of additives and probiotics the E. coli were exposed to, while the dependent variable would be the area covered by their resulting growth. The additives I decided upon were monosodium glutamate, Allura Red, a common red food dye, powdered caffeine, fine-grained table salt, and confectioner's sugar, all of which have supposed links to IBD flare-ups. The probiotics were Lactobacillus acidophilus, a common bacterial supplement. I took sterile petri dishes and added agar, crushed additives, and, in some cases, probiotics, in such a manner that all dishes were filled to the same volume. To allow for potential error, I made 2 petri dishes for each combination of additives and probiotics. I then streaked the plates with Escherichia coli, strain K12, the strain most common to the human digestive tract. Finally, I used sterile glass beads to roll the bacteria around, spreading them as evenly as possible over the surface of the plates, so that I could measure growth area easier. The dishes were then sealed and placed in an incubator. On a side note, the seal was not removed even after the dishes were autoclaved after the experiment was over. 2 days later, I removed the dishes from the incubator and took the following images for measurement purposes. I noted that only E. coli grew on the plates, and all growth had actually been complete since the first day. Here we can see an example of a dish laced with salt. A grid has been measured to scale and superimposed over the dish to allow a sample area marked by the blue box to be analyzed for E. coli coverage. Specific area was then determined by marking shapes over all areas occupied by E. coli, grouping these together to minimize remaining area, and finally estimating percentage coverage based on the coverage of this new hybrid shape. The sample area was not random, but rather was specifically chosen to include as few areas of disrupted surface, such as where salt or probiotic crystals might be displacing the agar, as possible. This is a fairly average example, but I did notice a few irregularities. To identify some of these irregularities, plates laced with caffeine showed almost no E. coli growth, and colonies on plates with MSG exhibited gelatinous, abnormal formations, as shown here. Otherwise, it is interesting to note that E. coli in dishes with probiotics tended to grow better than in dishes without, with the exception of the salt plates. Overall coverage was consistently within the range of 70 to 90% of the Petri dish, with the exception of caffeine, as aforementioned. Thus, I was able to conclude that my hypothesis held true for caffeine, and possibly MSG, that additives can indeed cause an imbalance in bacterial colony size. As for probiotics, it seems that they do indeed counteract irregularities in E. coli colony size. However, the case of the complete lack of E. coli in the caffeine dishes without probiotics is unsettling. One has to imagine the kind of damage caused by the human body vainly fighting itself over whatever bacteria grow to take over the E. coli's place. In the end, this experiment served to open the door to future planned experiments connecting various internal and external stressors to IBD.
Everything went very smoothly in this experiment, and I hope to be able to build upon this data by looking further into depth to find out more about modern digestive disease and new ideas for approaching it. Thank you.